Good morning everybody. I am Dr. Nitin Joshi, Professor in Urology with teaching experience of 25 years working as consultant neurosurgeon in Panvel. Today we will see one important immunology topic, and that is cell-mediated immunity with respect to the immunotherapy in cancer prostate, BCG vaccine, HIV infection, transplant rejection as well as genitourinary coughs. Cell-mediated immunity is a type of acquired immunity or adaptive immunity or the specific immunity. Now, acquired immunity means the ability of the individual's immune apparatus of fighting against a foreign antigen when he acquires only when he is exposed to this antigen in life, that is after birth. So this immunity is not since birth and so called as acquired immunity. The cell-mediated immunity, as name suggests, it involves various cells, particularly the T lymphocytes, natural killer cells, macrophages. It does not involve antibodies. In fetus, the lymphocytes develop from the hematopoietic stem cell, which is called as committed hematopoietic pluripotent stem cell, because it is committed only for development of the lymphocyte progenitor. The lymphocytes which have been destined to form T lymphocytes will leave bone marrow and will via blood will enter the thymus gland, which is a central lymphoid organ or primary lymphoid organ. In the thymus, these immature cells will proliferate without any prior antigenic stimulation. In this thymus gland, these immature lymphocytes, they will get trained, they will get educated and become immunologically competent due to presence of hormone thymosin. They are trained to identify or recognize the self antigen or own cells. They are trained to identify or recognize the non-self or foreign antigen, thus to differentiate between the self antigen and the non-self antigen. And the ability to recognize the foreign antigen is due to acquisition of special surface mechanism, and that is the T cell receptor. So there is acquisition of special T cell receptor to each cell. As a result, every T cell will be able to identify only one type of antigen or antigenic determinant called epitope. Then not only they will acquire the antigenic recognition mechanism, but they will acquire a specific surface molecule which is responsible for cluster of differentiation. The cluster of differentiation, that is T4 helper and T8 cluster. T4 helper also called inducer, while T8, that is the CD8, there are two types of cells, and that is cytotoxic T lymphocytes, or they are also called as T killer cells, which are important, the effector cells of cell mediated immunity, and T8 suppressor cells, also called as regulatory or inhibitory T cells. Once this they acquire, then they become immunologically competent. Immunologically competent, that is when the specific T cell is stimulated by an appropriate antigen, gets stimulated so that this blast transformation, clonal proliferation and differentiation into effector cells so as to bring about immune response. Once they are trained, then they leave the primary lymphoid organ, that is thymus, and then through blood they are carried to settle into the secondary lymphoid organ or peripheral lymphoid organ like the spleen, lymph node, bone marrow, mucosa associated lymphoid tissue or gut associated lymphoid tissue, so MALT or GALT. Now whenever there is entry of antigen in the tissue or circulation, then in case of tissue, from the tissue it will come out in the tissue spaces. In the tissue spaces start the fine lymphatics. These lymphatics drain the lymph. So along with the lymph, they will be draining this particulate foreign antigen to the draining lymph nodes. Draining lymph nodes contain antigen presenting cell as well as lymphocytes, which are the two important cell types of acquired immunity. If there is entry of the antigen in the circulation, then the spleen is the filter of blood, and so the foreign antigen is captured and is filtered by the spleen, which also contains the antigen presenting cell and the lymphocytes. Antigen is any substance which, when introduced in the circulation, is responsible for stimulation of immune apparatus to produce certain cells or molecules which react with that particular antigen specifically so as to neutralize the antigen and ultimately eradicate the antigen from the body. Antigenicity is the ability or capacity of the antigen to induce the immune apparatus to synthesize the molecules and cells which will react with the specific antigen to eradicate the antigen from the circulation. Antigenicity can be enhanced by combining the antigen with certain adjuvant. For example, in vaccine Sipulsi T for the cancer prostate, the antigen PAP, that prostatic acid phosphatase, can be combined with the adjuvant, the GM, that is granulocyte monocyte colony stimulating factor. In case of acquired immunity, two types of cells which are important are antigen presenting cells and lymphocytes. Antigen presenting cells are the important cells which are able to capture antigen, process the antigen to release the antigenic determinant. That is, majority of the antigens are complex protein in nature, and so they are digested by these antigen presenting cells. The three main important antigen presenting cells are the dendritic cells, macrophages and B cells. The professional antigen presenting cells are dendritic cells and macrophages. The protein is digested by this antigen presenting cell to form small peptide molecule. The small peptide molecule then moves to the surface of antigen presenting cell. Then this small peptide molecule then combines with the major histocompatibility complex type 2 surface molecule of the antigen presenting cell. Now this antigen presenting cell then represent the antigen to the specific T cell having specific T cell receptor with respect to the specific major histocompatibility complex type 2 surface molecule of T4 helper cell. Because of presentation of this antigen, now there will be stimulation of the T4 helper cell resulting into clonal proliferation, blast transformation and differentiation. T4 helper cells get differentiated into two types of subpopulation, that is T4 helper type 1 and T4 helper type 2 subpopulation. These two subpopulations depend upon the type of cytokine secreted by T4 helper cells as well as the environment of the cytokine in which they develop. That is, for the development of type 1 T4 helper cell and the type 2 T4 helper cell, different types of cytokines are required. Gamma interferon, interleukin 12 and interleukin 18, these cytokines are required for development of type 1 T4 helper cell, while interleukin 4 is responsible for development of type 2 T4 helper cell. Now, whenever there is antigenic stimulation of immunologically competent T4 helper cell which was not yet stimulated by antigen, now when stimulated by antigen presenting cell, because of the specific antigenic determinant with respect to major histocompatibility type 2 surface molecule of the antigen presenting cell to the specific T cell receptor of T4 helper cell with respect to the major histocompatibility surface 2 molecule of the T4. Now there is a release of gamma interferon. The released gamma interferon is by the stimulated T4 cells as well as natural killer cells. The released gamma interferon will do two things. One, that is inhibition of expansion of type 2 helper T4 cells, and they will stimulate the dendritic cells and macrophages. Stimulated dendritic cells and macrophages is responsible for secretion of interleukin 12, which plays a key role in development of T4 helper type 1. Developing T4 helper cell type 1 will release more and more gamma interferon. The developed mature T4 helper type 1 will release not only gamma interferon but interleukin 2 as well as tumor necrosis factor alpha. The gamma interferon and the interleukin 2 is responsible to bring about the effective effector element of cell-mediated immunity. The effective effectors of cell-mediated immunity are cytotoxic T lymphocytes, macrophages and natural killer cells. Cytotoxic T lymphocytes is having the CD8 surface molecule on the surface, and it works with major histocompatibility class 1 surface molecule. This is responsible for targeting the target cells and responsible for direct lysis and the destruction of targeted cells by release of the lysosomal enzymes and further release of tumor necrosis factor cytokine. The next effector is macrophage. As compared to polymorphonuclear cell, macrophage is effective phagocyte responsible for phagocytosis, that is, it engulf, ingest, digest and destroy the foreign antigen. Though it is effective in the various pathogens for phagocytosis and killing, there are certain bacteria which, like tuberculosis, leprosy, brucellosis, typhoid, which resist intracellular killing. And so when the macrophage phagocytoses this bacteria and wandering macrophage transport or travel from one place to another, there is transport of these bacteria from one place to another, and so phagocyte and phagocytosis is responsible for the disease spread. But when this macrophage phagocytoses these bacteria, the macrophage is activated, responsible for release of macrophage aggregating factor, macrophage activating factor, and therefore conversion of macrophage into effective effector elements like epithelioid cells, giant cells, particularly Langhans giant cells in tuberculosis. So that the developed effector cell has got more efficiency, because one single macrophage is not able to capture too big foreign molecule, then such giant cell or enlarged effector element can capture the foreign antigen. The natural killer cell or the non-T, non-B cell, the T lymphocytes and B lymphocytes have been classified depending upon the presence of specific surface molecule, which is an antigen recognition mechanism. That is, for the T cell, there is T cell receptor, while for the B cell, there is immunoglobulin-like molecule which acts as a receptor for the recognition of antigen. When the lymphocytes have neither T cell receptor or immunoglobulin, they are called non-T, non-B cells. These lymphocytes, like other lymphocytes, are developed from the pluripotent hemopoietic stem cell in the bone marrow. However, there is no maturation of these lymphocytes in bone marrow or thymus. Also, their cytotoxicity is not the antibody dependent or major histocompatibility restricted. It doesn't require prior antigenic stimulation. It belongs to the natural or innate or inborn immunity. It is not a phagocytic cell, but it contains certain granules. Granules contain hydrolytic enzyme like perforins. Perforins are responsible for development of transmembrane holes or pores of the target cell. Through these pores or holes, there is entry of cytolytic and cytotoxic components into the targeted cell, causing the lysis of the cell. Natural killer cell has got important function in the immune surveillance, because that is in cancer immune surveillance, because it is able to detect any mutation in the cell, and also it has got important function in tumoricidal, and therefore it plays a role in cancer therapy. Now the concept of what is called T cell checkpoint, that is when the T cell have achieved their desired function of elimination of the foreign antigen, then the function of T cells should be inhibited so as to prevent the destruction of own cells. This happens that whenever there is cytotoxic T lymphocytes are stimulated, they are responsible for release of cytotoxic T lymphocyte associated antigens. These cytotoxic T lymphocyte associated antigen stimulate the T8 suppressor cells. The T8 suppressor cells, also called as T regulatory or inhibitory cells, they inhibit not only function of cytotoxic T lymphocytes but also T lymphocytes as well as B lymphocytes. So a sort of immunosuppression which is important for the prevention of destruction of our own cells. Now the memory cells, after the desired effect of elimination of foreign antigen by the cytotoxic T lymphocyte, natural killer cells or macrophages is achieved, then certain T lymphocytes, rather than entering the general circulation, may enter the lymphoid organ like the spleen and lymph node. They retain the previous experience. The memory of this previous experience is retained, and therefore they are called as the prime cells or memory cells. The lifespan of these memory cells is much longer than the effector cells. So when this antigen, same antigen enters the body or circulation second time, then the memory cells are responsible for quick, rapid response at the site of entry. They recognize the antigen at the site of entry, and so they react with the antigen at the site of entry, thereby preventing the spread of the infection further. And so there is heightened, strong, immediate response to this antigen which has entered second time due to presence of memory cells is called as secondary response. The cell-mediated immunity plays important role in the one that is cancer surveillance, in cancer immunity, transplant immunity, and the protection by the phagocytosis of certain bacteria like tuberculosis, leprosy, brucellosis, then fungi like blastomycosis, coccidioidomycosis, as well as the trypanosomiasis like protozoa. And so in HIV, when there is a complete collapse of cell-mediated immunity, they are responsible for development of unusual infections and malignancies. Thank you very much.